We wish we were there with you in person.
You should have come. But thank you so much for being here. We have an audience in the room of about 20 of us, and we have the audience in Zoom that you can see. So without further ado, please get us even more excited about biodesign and all the things that Stanford has done to make it such a good and excellent model for us to be inspired to help bring solutions to the healthcare industry and the people.
All right. Well, thank you so much, David. It's a pleasure to participate. And again, we wish we were there with you in person. Yusuf is a hard act to follow, but we'll do our best to keep the energy high and share some good information that hopefully will inspire more good work from the group down there. So I'm going to share my screen. Bear with me one moment. Are you guys seeing? Oops, nope, I'm not going to share it. I'm going to slideshow it. That's what I want.
We are starting to see. Yes, Lynn, we are.
Yeah, looks good for me.
All right. You'd think after years on Zoom, this would all be a little bit more natural and quick. But anyway, here we are. All right. So buenos dias, everyone. We're delighted to participate. We are going to, Ross Venook is here with me, who's leads many of our programs at Biodesign, including our senior bioengineering capstone course. And Paola Calvachi also is here. She is a recent graduate of our full-time innovation fellowship program. And so what Ross and I tell you will maybe feel a bit academic, but what Pao tells you is the real, where the rubber hits the road. She's out there doing biodesign and will share some of her story with you today. We're going to divide our presentation today into 3 sections. I'll tell you a little bit about Stanford Biodesign and the fundamental biodesign innovation process that we teach. Pao will share with you how it really works on an innovation project. And then Ross will talk a little bit more about how you can teach biodesign and related principles in your courses and programs. So first, very quickly, Stanford Biodesign was founded in 2000 by Paul Yock, and Josh Macauer came on as our first fellowship director and co-founder of the program. The whole idea behind biodesign was really Paul was a young innovator who, according to Paul, who's rather humble, got very, very lucky that he had the benefit of good mentors when he was a young inventor trying to bring technologies into patient care. And he started to think about, how can we provide that sort of mentorship to more aspiring innovators on a broader scale so that, more technologies can reach patient care. And so this was really the whole kind of fundamental principle behind creating an educational unit that would be focused on preparing and training innovators, giving them a repeatable process for innovation that they could practice and improve out over their careers. Here is our mission and our purpose and values today is advancing health outcomes and equity through innovation, education, translation, and policy. And we do this really in 3 primary ways, through teaching, mentoring, and also seed grants that can help early stage projects get from a great idea actually to commercial funding. What we teach, which I'll go into in a little more detail, is this 3-phase process for identifying compelling unmet healthcare needs, inventing new technologies to address them, and then preparing to implement those into patient care. We teach fellows at Stanford, Stanford faculty, Stanford students, of course. We support researchers through our seed grants. We have global training programs for faculty and fellows around the world, and also an executive education program. We reach about 400 to 500 different trainees per year. So far, there have been over 50 companies who've come out, who have launched, have come out based on ideas initiated during training, with many others that have been launched by innovators using the process after training. And this is really where the kind of the stat we're most proud of. More than 7.6 million people have been helped by those 50 companies or so, with the numbers, as you can see, growing rapidly as more of these technologies make it into patient care. But our real product is the people that we train. And we have nearly 200 graduates of our full-time fellowship program at this point. And this network effect is fantastic in terms of all of the people we've trained who now pay back to bring up the next generation of innovators and become part of this lovely community. So let me tell you a little bit more about the process that we teach. Especially in universities, there's a lot of technology innovation that happens with this kind of tech push model. So someone makes a fascinating discovery and then goes looking for a problem that it can be used to solve. And it can be a great way. I mean, many important technologies have come forward using this model. However, particularly in healthcare, where it takes so much time and money to bring something all the way from an idea to patients, we think that there's maybe a more effective and efficient model, which is basically to start by identifying and focusing on a most compelling unmet need, become an expert in that need, and then once you're certain that you understand what's most required, invent something uniquely designed to solve that problem. And we call this need pull or need-driven innovation. It is fundamentally based on the design thinking process, or user-centered design is another word that term you may have heard. But design thinking really grew up in sort of the consumer product space, where it's the same person who makes the decision to purchase something, pays for that purchase, and then uses that purchase. But as all of you know far too well, that's just not the way it works in healthcare, right? We often say healthcare is this 8-headed monster, or maybe there are even more heads at this point. But anyway, you have to think about the concerns of the patient and the physician, the facilities where care is delivered, the regulatory agencies that oversee activity in the space, the payers, both private and public payers, the press and professional societies can have quite an influential role. And then there's the government, right, where sometimes it can feel like a pendulum swinging in terms of how easy or difficult it is to advance new technologies. And so to deal with this, we came up with a domain-specific version of design thinking that we call, again, the Biodesign innovation process. And we'll just very quickly go through what the steps are in the process. So first, and the secret sauce really is what we call need finding. As Yusuf said, choosing the right project to work on is that unsung hero. It is the secret sauce, and it is where you should spend a substantial part of your time, both as an innovator and as an educator as you're interacting with your students. So we like to think that the best way to find a good need is to start by identifying many needs. We put our fellows together, using them as an example, on interdisciplinary teams, people from different backgrounds, spanning engineering, medicine, and business. And then we task them to go out and find, at least 100 different needs to start with. And they characterize each of those needs with a need statement. A need statement has a very specific construction, basically a way to address a problem in a well-defined population in order to achieve a desired outcome. So that might look something like a way to rapidly diagnose patients with skin lesions in order to accelerate time to treatment. The key thing to understand at this point is that the need statement is a hypothesis, right? So it's your best guess based on observations about what you think is going on and what is most needed or most lacking in that space. And then your job will be to go forward and either prove, disprove, or refine that hypothesis over time. And as that gets more refined, it becomes sort of like the mission statement for the project. Another key thing here is at this point, no solutions are allowed. This upfront need finding and need characterization should be completely solution independent, so that you keep your broad, you don't limit yourself, right? If you start thinking about needs in terms of like, oh, we need a better catheter, or we need a more efficient, a more effective stent, you're limiting your creativity and what you can accomplish the best, and the best way to accomplish what's most needed. We've sort of said this, but if you come up with solution ideas at this stage, the best thing to do is jot them down, set them aside, and carry forth with your need characterization process. You'll come back to them at a later time. Once you have lots of needs identified with this kind of preliminary need statement, what you want to do then is essentially have those needs compete for your time and attention. So through progressively in-depth need research in these 4 areas, you're going to become increasingly expert on your needs, and they'll be competing. So at first, you'll see, well, let me quickly say, the 4 key areas to look at this stage are the disease state fundamentals, especially making sure you really understand the mechanism of what's happening, understanding the existing solution landscape, not just in devices, but across the entire spectrum of solutions. Again, we're not necessarily going to presume that a device is what's most needed, right? We want to find the best solution for the need. You also will do this stakeholder analysis to understand the interests of all of those different stakeholders that make up this 8-headed monster, and then market analysis, not necessarily because we want to get rich, but because we need to make sure that there's enough of an opportunity that we can raise the capital that we need to develop and commercialize the technology, and so it actually gets to patients. Then with this progressively detailed information, we'll go from hundreds of needs down to dozens of needs, and ultimately to the top few needs that you want to take forward and that you're going to invest your time in. So this is a very objective way, rather than just relying on gut instinct to tell you, with placing a bet on where you're going to spend the next 5 or 10 years of your life. For the top needs that you decide to take forward, the way that you make all that need research actionable is to come up with what we call need criteria. And need criteria are the requirements that whatever solution you invent must meet in order to have a shot at getting adoption, right? So you take everything that you've learned through your need research and you come up with these basically 2 kinds of need criteria. The must-haves are the things that absolutely must do in order to be able to display standard of care. And then there's the nice-to-haves, the things that would really sweeten the deal and get not just the decision makers interested, but also kind of the influencers who can support adoption of the criteria of the solution. So basically the must-haves are it's a threshold effect. You have to meet them or you're not going to succeed. Nice-to-haves provide kind of what we think of as market differentiation. Need criteria fall into 4 general categories: efficacy, cost, safety, and usability. And you should really be thinking broadly about having criteria in each of these zones. We also like to think about access, but access can kind of cut across these, and decisions you make, for instance, about cost will affect patient access, usability, things like that. We also want to be actively thinking about how what we need to do to make sure that our technology can reach and benefit the most people possible, particularly those who might currently be underserved. So here's an example of what need criteria look like. You can see that they are specific. They should be associated with some sort of a quantitative benchmark that you can use as a target. That's hard to do at first because you don't have perfect information, but you can, based on your research, make your best guess. Something at the point of care would need to take less than 10 minutes because that's the average time of a dermatology visit, et cetera. Current tools are this accurate. We need to be as good or better. Those are the kinds of, inexpensive. Currently, biopsy is around $190. How can we come in at or below that in order to get to drive adoption? So you can see how these need criteria work. That's your launching point for concept generation when we move into the invent phase of the process. And we like to say, given enough time, sugar, and caffeine, you will invent something great. The way that you use the need criteria there is to first use them as a launching point for ideation. And then they're your initial screen You'll come up with, dozens or hundreds of different ideas. The very first screen is does each idea have a shot of satisfying the need criteria? If not, you're going to set it aside, or you're going to go back to brainstorming and reimagine that idea until you think it has a shot at meeting those need criteria. What you'll do then, once you've whittled down to the ones that look good against the need criteria, is you'll start a more rigorous concept screening process. Again, making your solution ideas compete against one another for your time and attention. So as I said, first you do that against your need criteria, and then you're getting progressively more in depth around intellectual property, regulatory pathway, reimbursement or payment, business models, and of course, technical feasibility. So this is an iterative screening process where, again, you're going from many ideas down to the one that has the best shot of satisfying the need and making a difference in patient care. The implement stage is where the rubber hits the road, and you're really going to be looking at weaving together a comprehensive strategy around IP, R&D, clinical, regulatory, quality, payment, market, sales and distribution, as well as your competitive differentiation and business strategy. You also need to think about, of course, the best pathway to reach the market. Not every good idea is a startup, right? You may be able to license or partner in order to bring something forward. You'll have to think about how much time and money you're going to need, where that money will come from, and the story that you'll tell to raise the support you need, whether through investors, your employees, and other supporters. So that was a very quick overview of how the process works, but all of this is going to make much more sense when you hear it through an example. So Pao, I will turn over to you at this point.
Thank you, Lynn, and hello, everybody. Hola a todos los que están allá en Bogotá, un saludo especial. So as Lynn mentioned, my name is Paola Calvachi. I just finished the Biodesign program, and today I'll talk about my experience as a fellow. For this, I'll walk you through my team's process using a case study. So next slide, please. Well, as Lynn mentioned, the team is one of the most critical parts of this journey. In my particular case, we were a team of 4 with all different backgrounds. The backgrounds were like in the industry and academic experiences. For instance, Stacy had a background in medical device development and organizational management. Kevin, he had a background in biomedical engineering and a PhD in stroke and rehab. Ankit, he has a background in medicine and he is doing or completing his fellowship in cardiology. And me, I have a background in medicine and also bioinformatics. Next slide, please. Well, during our year as a fellow, the area of focus was cardiology. We did our clinical immersion in 3 different hospitals around the area. 2 of them were community hospitals and 1 of them was academic. At the end of our observational period, we got 464 needs statements that were filtered down to 3 during the first 5 months. And then during the last month, in order to complete 6 months, we filtered down those 3 to 1. Next slide, please. And in order for us to analyze and collect information for each need, we conducted more than 100+ interviews with the stakeholders, with several stakeholders, launched more than 4 surveys to collect patient information and experiences. We developed many prototypes and learned how to perform an economic evaluation of the healthcare interventions using Markov models. Next slide, please. So my team process to filter down the needs consisted in evaluating the needs separately, gather information as much as we can from different sources as possible, finding insights into the problem, and identified pretty early on what are the killer risks. So here's an example of our top 3 needs. We have a variety of incremental, mixed, and blue sky type of needs in different areas around cardiology. So we had cardiac surgery, electrophysiology, and general cardiology. The insights that we identified tried to give us a different perspective on how can we approach the problem or the need from another angle. They were related to the mechanism of action, as you can see on the first one, or like what the procedure that it's available, it's not current available for everyone. Or in the third one, it was related with treatment and diagnosis. diagnosis, sorry. Next slide, please. And for this particular case study, I will focus on the problem we observed multiple times during our clinical immersion. There was an increased number of patients during developing cardiac arrhythmias after cardiac surgery. Basically, we identified that the primary mechanism of action associated with that problem was atrial irritation caused by a pericardial fluid that was retained in the posterior part of the heart after these interventions. The population, as you can see underneath the statement, is pretty broad. And we had many iterations on the population, but at the end, we decided that for this particular case, we wanted to be very broad because the mechanism of action was like very comparable among all the patients that were having this problem. And for the outcome, we basically needed to have something that was measurable, that we could measure with clinical trials. And for that, we identified as the incidence of heart arrhythmias during the first 3 days after the surgery. Next slide, please. Okay, so after identifying the problem and the mechanism of action, we generate the needs criteria using 4 main categories, as you can see on the slide. So we use performance, usability, safety, and value. For each category, we came up with a must-have and the nice-have criteria and a way to associate it with a benchmark, as you can see on the 3rd column. The performance criteria had to be associated with the outcome, in this case, the incidence of atrial fibrillation after cardiac surgery. And the usability and the safety criteria came from the user requirements that we obtained after conducted multiple interviews and the surveys with the patients. The value, it came from the health economic value and reimbursement analysis we conducted. And also we had for each individual need, the need type, the mechanism of action, and the insight, so we can compare and contrast among them all the time. Next slide, please. Well, in parallel and as part of the ideation process, we started to organize like many brainstorming sessions to generate multiple ideas, and we only took the prototyping phase, the ones that make sense. And based on our risk analysis and research, we were constantly evaluating with a color scale different matrices. Here we also evaluate the disease state, the stakeholders, all the information that we got from them. For the stakeholders, as I mentioned before, we conducted more than 100 interviews, but those interviews were across the world because basically we wanted to have like a broader opinion on the same project and on the same problem. The market opportunity was calculated based on the number of surgeries performed in the United States. We based those analyses only in the United States mainly, but we also had numbers across like around the world. The competitive landscape was just to give us more information around like what are the gaps that we need to fill with our solution and how can we do that, and the health economic values just to create impact on the patients and the hospitals and all the stakeholders involved in this problem. Next slide, please. So based on our old research and the risk assessment, we were constantly evaluating, as I mentioned before, with a color scale, a matrix with all the components from the problem, the ideation, and the implementation phase. We selected 3 colors for this to evaluate each category. So the red color that you see on the screen was to show there was a red flag associated with that particular category. The green showed that there was like an opportunity or a path to achieve our goal. And the yellow one was there might be some risk, but either we didn't have enough information or the information we collected was conflicted and we have like a big uncertainty of that. So we needed to have more information in order for us to make a decision on put it as a green or as a red. Next slide, please. Okay, and as we were doing that, at the same time, we were starting the ideation phase with the brainstorming process. And for it, basically, our process consisted on generating a matrix, as you can see on the first picture on the left. So we had idea generation for that. And for that particular phase, we came up with different how might we questions. The how might we question target the problem that we wanted to solve. So for instance, in this case, The how might we question was like, how might we reduce the atrial irritation that these patients were having after cardiac surgery? That was one how might we. Another one was, how might we realize if the patient was having atrial fibrillation at early on? And with all of them, we came up with multiple ideas. Once we have the board filled with sticky notes, we jump into the next part that was idea sorting. And with the idea sorting, basically we had this chart that you can see where we have the feasibility versus if the concept meets the need or not. And we only took the ideas that were on the top right corner. With those ideas, we moved to the third phase, that was the needs criteria and winner circle. We call it that way because basically we took each of the needs and we put it in the most have criteria. As Lynn mentioned, every single concept has to fulfill all the most have criteria, otherwise we need to kill the concept. So we started with one concept, we put it on the performance. If they fulfill those criteria, then it moved to the usability, and if fulfill it, like you just like go on and on. But some of them, or I would say most of them, died on the performance and usability part, and only a few of them made it to the winner's circle. Only the ones who made it to the winner's circle passed to the next phase, that was prototyping. Next slide, please. So for the three concepts that that made it through the winner's circle, were something like the three concepts that made it to the winner's circle were continuous flushing system, and a spider tube plus a posterior pericardiotomy and a pericardial clip suture. Next slide, please. With these concepts, basically what we did was to create like two different prototypes. So the first one was a looks like kind of prototype, and the second one was a works like prototypes. And we also prototyped the problem. Why? Because we wanted to have like a better understanding of like, why do we have the fluid stacking on or anesthesis on the posterior part of the heart, and how would be the best way for us to access it. So that's the first picture that you see on the screen, and it's basically a plastic bag with a pig's heart in the middle. We put some liquid behind that was water, and then we just tried to come up with a cannula to extract the fluid, and that gave us a lot of ideas. Next slide, please. So with that in mind, the concepts were improving over time, and with our leading concept in this case was the spider tube plus the posterior pericardiotomy, we started to evaluate the implementation phase. And for this, we had in mind the IP, the regulatory, and the reimbursement strategy and the landscape. In this phase, we submitted different patents for the concept. We found that they have freedom to operate, and formulated the milestones that we need to achieve in order to have the regulatory approval from the FDA, and how can we get reimbursement at the end. Next slide, please. So after assessing each individual need, we compared the progress of each of them and killed the ones we identified killed the risk or could not move forward at the same speed as the other. Ultimately, what happened with this particular need, the one that I mentioned you, the posterior pericardiotomy one, was that a new technique emerged, and that new technique was pretty feasible to perform for all the cardiac surgeons. When we were talking to the stakeholders, they were basically pretty excited about the results that new technique was having. It was cheap. They didn't have to pay anything for it. So it wasn't like a medical device. It didn't have all the costs associated with that. And at the same time, it was pretty safe for the patients. So at that point, we realized, I mean, we could create something to come up with a medical device to solve the problem, but they already have something that was even better from what we were proposing. So we killed the risk and we move forward with another one. Next slide, please. And looking at the process retrospectively, I can say we have spent endless hours together going in circles. That's pretty normal. Dealing with uncertainty, that's also pretty normal. Learning about how to make decisions with incomplete information. Brainstorming, researching, and talking to people around the world to hear different approaches on like how they might treat the problems differently. And as you saw in my case, my team and I followed the Biodesign process. But at the same time, we created different tools to help us out to choose between one need or the other. And those tools might be different from team to team because the process gives you the freedom to choose whatever is most relevant for you and whatever makes more sense for you and your team. Next slide, please. And with this, I will hand this to Ross to continue with the teaching biodesign process.
Thank you, Paola. And can people hear me okay? Okay,
Yes.
I'm seeing some nods, so I appreciate that. My slides, there's kind of a lot of information on them, so I'm going to post in the chat here. This should be a link to a PDF that everybody can see, so that you can sort of have your own version in case that's useful. And if you have trouble getting to that, just let me know. You know, what Paola went through, and thanks, Len, for advancing the slides. What Paola went through is, of course, the fellows experience, a group working full-time around the clock. We have a lot of different sort of flavors of educational experiences within biodesign. And so, and I know that a lot of the people at this meeting are going to be leading more sort of courses rather than full-year, full-time experiences. So I'm going to talk a little bit about that, share what we do in our capstone course at Stanford, just very specifically as an example, along with some, you know, I guess, challenges that we typically face, as well as you know, recommendations, tips and tricks, and have some time for Q&A, and look forward to hearing specific questions. So next slide, just to set the table. You know, as Pao was talking about sort of this full-time fellowship, which I would put in the category of practical training at Biodesign, which we also lead a faculty fellowship. I spend most of my time working with people in the traditional course structure, so either graduate or undergraduate or some mixture of students. Some of them are single discipline, so the courses I lead are all bioengineering students. But there are also some versions where we have people from different disciplines. Our grad course, for example, brings together people from the school, the business school, medical school, and engineering school. We also lead some seminars and things like that. What these all have in common is that they're all open-ended and solution agnostic. They all involve teams of students working together. They all follow a design process, and the focus depends upon the specific course. Of course, we all use the biodesign process, and they all involve some combination of a truly unmet need as well as a solution. And next slide, just to give you, we're going to zoom in on one of these courses in particular, the capstone course that I lead, which hopefully will give you an opportunity to think about it critically and ask questions related to that. So for background, we're on the quarter system at Stanford, so it's sort of 10 weeks per quarter. We have autumn, winter, spring. Most of our biodesign class sizes are in the 20 to 40 students range. So I know that some of you may be working with smaller groups, some of you may be working with much bigger groups, but that's kind of what we tend to address in terms of our courses. And so we usually have between 4 and 8 teams, depending on the situation. Next slide, please. So the example course I'm going to dive into is a 2-quarter course. It's designed for seniors in bioengineering. And again, we call it our capstone course. We follow the biodesign process, the identify, invent, and implement. And the way that it works for the first quarter in our fall quarter is sort of as you see here. We do a little bit of identify, a little bit of invent, and a little bit of implement. Now, this being an engineering course, we have the second quarter that's spent mostly doing building and testing, which I'll get to. The goals for the first quarter are to kind of race through the needs finding, needs definition, all the same steps that Lynn described and that Pao described over the course of about until, let's see, our students define their concepts sort of in week 6 or 7 of the course. And this is an accelerated time frame, and I'll talk about why that is. The goal by the end of this first quarter is for them to have a needs statement and a set of need criteria that are solid. a concept that they can move forward with, and then an execution plan for the second quarter. Next slide, please. And again, the purpose of these slides is to show you just very practically, like, how do we do it in the course that we run. And I'm happy to share our syllabus, happy to share our materials. Feel free to reach out and we can share those things. In the winter, we have the students, the next slide, please. They have established a project plan toward the end of the first quarter where they establish their own milestones. And we have them establish at least 4 milestones. Most teams choose 4, but some choose 5 or 6 milestones that they're going to hit over the course of the second quarter. And what we do, as you can see here, is we sort of layer in a little bit of IP strategy, a little bit of regulatory analysis, a little bit more about commercialization. But the real focus of this course is on engineering. And so it's again, building, testing, modeling, technical milestones typically in this course. Next slide. So how does the fall work? And this is something that we've evolved over several years. And I'll just, again, go into details so that you can kind of see how it really works in our class. So by week, we got 10 weeks. And each week, there's sort of a different focus on a different aspect of the process. And so when I say process material here, what I mean is this is the sort of the learning that the students are doing in that week, where maybe we're having a workshop or a lecture session with a guest or something like that, that focuses on this topic. Next slide. We also have this as a lab course. And so, we have some didactic sessions each week, or sort of one shorter didactic session each week. For us, it's Monday morning. And then in the Wednesday and Friday afternoons, they're in the lab for a few hours each of those intervals, and they're doing project work mostly. So we do a little bit of workshopping, and then turn the time over to the student teams to work. So that's sort of indicated in yellow here. And what they're working on is the stuff that we had been talking about over the last week. And so they've got a deliverable related to that each week. So in the first week, they're choosing their need area. In the second week, they have to deliver a short list of need statements. The third week, they have to have need criteria well developed and need research done, et cetera. So on that third line there. And so by week 7, they're presenting on a coherent need that they've decided to pursue, as well as the concept that they think they want to push forward. Next slide. A couple of things, yeah, and so you can see it's kind of layered like this, right? So they're, we're talking about it, they're doing a workshop on it, maybe a worked example in week 1, and then they're delivering their artifact about their project in week 2. Almost everything in our course is about their project. So wherever possible, they're working on their project. Sometimes we have other examples, depending upon whether what they're working on really ought to be done in a trial version before they apply it to their project. It kind of depends. All right, next slide. So the other thing that we layer on here is, and in our classes, it's on Wednesday mornings. Wednesday mornings, we're having mentoring meetings. I'll say that these are what I think are kind of the minimum mentoring meetings that we have with our teams. But in point of fact, we've actually gone to meeting with them every single week. And a mentoring meeting would be maybe a TA and or faculty member with the team. So sitting down, talking about their project and talking about their process. And that separation is something that I'll get back to, I think is really important. At least it's been important in our work. So we have these mentoring intervals. And if you hit the next slide, one thing that we do, our students don't go into the clinic. They do meet with clinicians. So each project has a dedicated clinical mentor that the students can reach out to. And we like to have a minimum ask for our clinicians and allow them to give more on the back end. The minimum ask for this course is that the clinicians meet with the students twice. That's it. And if they meet at the right time and the students are well prepared and the clinicians are well prepared, then that has worked for us quite well. Some clinicians and teams end up working together more, but really it's at this, when the students have thought of their need statements and some of their need criteria, and they run their top 3 by the clinician, and the clinician's able to give the kind of input that only a clinician can give, kinds of things that the other mentors, myself and the TAs really can't give them. And so we have an additional meeting that the students have with the clinicians in weeks 3 and week 6, when they're deciding on what need to pursue and their specific need language, and then when they're deciding on their concept. So that's what our first quarter looks like. The students describe it, well, they basically say it moves way too fast, and it's really hard to do all this stuff in this time frame. And my perspective, we used to take the whole quarter to do this part, and we decided to accelerate it and to actually get them working on their project plan and trying to do experimental work. You can see that there's some milestone 1 work by week 10. We decided to do that for a couple of reasons. The most important was that if they finish the first quarter and they haven't done any experimental work, typically what happens is that when they start up the second quarter, the first experimental stuff takes a while to get started, and then it doesn't work, and then the quarter's over. So we like to accelerate the early part to give them a chance to try some things, have those things not work out, and then they can pivot. They can find their way by either going back to the need criteria or going back through the process. So that's why we do it in such an accelerated fashion. All right, next slide. So as I was just mentioning, sort of some notes on this, it probably moves too fast, although I think anybody who's tried to do the biodesign process, Paola will tell you that it was also too fast for them. There's never enough time. And so we've found that this is a pace that can work for us and our students. You just have to change the expectations a little bit in terms of depth. And I think enabling those pivots really makes a lot of sense. And I find that the students don't really know what the next steps are until they try them. And so even if they're trying them in a little bit of a rushed fashion, once they go through them once, then it's like, oh, okay, now I get it. And then they're able to go back and rework and improve along the way. So that's how we do it in our class. Some of the keys to success, we'll talk about this a little bit later, is just making the class time be work time. My experience is that the more time that the students spend together working on their projects, the more they learn, the better the projects are. And so we try to get out of their way as much as possible. And so I do a lot of lurking, kind of like standing around watching them work and kind of, trying to get involved and, as little as possible, I guess, is what I would say. Other keys, I think I brought up this weekly mentoring, sort of having a cadence where they're talking critically about their project once a week. I've found that to be really helpful. And then, as I mentioned, sort of the timing of the clinical mentoring, as long as you get that timing right, then it can usually be quite efficient. All right, next slide. What I'll encourage people to do is to put questions into the chat as we go, because we started a little bit late. We're, of course, always running a little bit short on time. So I'd love to see those questions popping up. And then if it makes sense for me to address it right away, I will. Otherwise, we'll just kind of save them to the end. So please do throw questions into the chat, big, small, practical, blue sky questions. So I went through and described, kind of more specifically the fall quarter. What's the winter quarter about? So next slide. In our class, as I mentioned, it's really an engineering course. So the first quarter is to kind of get them to the starting line, if you will, on the technical aspects. And the goals for the second quarter, and thank you for putting in the questions. I love that you're doing that. Second quarter, what it's about is getting proof of concept data for their chosen concept. So that's really our aim, is that every team has some proof of concept data. Importantly, it doesn't have to prove it will work. It could prove that it doesn't work, right? But they have to have some data that allows them to make some decision about whether it's going to work or not work. So, what we do is we either, depends on the team. And at this point, the teams start to diverge, right? Because they're all working on different projects. All the projects have very different flavors. And so our first quarter is highly structured. We're all following the same process, same weeks. Second quarter is all over the map. Every team is doing different stuff all the time. And ideally, they're becoming more independent. I like to say, by the end of the second quarter, they shouldn't need us at all. Like, we're just there to kind of watch and cheerlead and, see if we can help them find resources. All right, and again, yeah, thank you for these questions. I will get to these as we go along. Let me finish up the last couple of slides, and then we'll have a little bit of Q&A time as well. All right, good. And so, yeah, I just want to emphasize that there are lots of different ways to run this. The one that I presented is one very specific flavor. We, in our course, don't focus a lot on the implementation part because of the increased focus on the technical work. Other courses, like our grad course, they're much more focused on getting to a business plan. How would this actually launch commercially? And they do less technical work. We do some prototyping, but not as much. So, those things can be adjusted depending upon what your, what your learning goals are for your students in the particular course. Our grad level course also, we involve a lot more non-Stanford community members. So the projects are a little more actionable, and so we bring in people from the community to interact with those teams of students. So the grad level course runs differently. It's a similar basic process, but those are some differences. And happy to talk offline about the specifics for that course. And then we, again, have a bunch of other flavors of courses. One that I'm particularly excited about, which I'll mention, is just a seminar that we've done where we do 4 full biodesign processes all the way to prototyping over the course of 10 weeks. And that's a fun one that we do with our sophomore level students. It's a different way to do it. So talking about the challenges very briefly. So there are a bunch of challenges that many of you, I'm sure, have faced. The biggest challenges that I think that we've started to address well in our capstone are around teamwork. And we have very specific tools for that. And I'm going to introduce a resource, the Student Guide to Biodesign, which is an online free resource that has an intentional teamwork module that I would encourage you all to look at. That's made a big difference in our course. We also make teamwork one of our top-line learning goals for the class. Other problems that students face around motivation. So motivation is really a combination of what value they think they're going to get out of the course and how likely they think they are to succeed in that. And so we have to work on both of those issues. A lot of this is about, in my experience, is about personal relationships with the students and figuring out what they need and what they wish to get from the course and how we can make sure that the course can bring that to them. A big challenge is students want to finish the project. And so we spend a lot of our time explaining to them that nothing worth doing is going to be done in 20 weeks. And but anything can be started. So I would say that framing has been really important on the challenges side for our students. The other challenges students face are around uncertainty and ambiguity. Right? The truth is that we as educators or instructors don't actually know the right answer. There is no right answer for most of these, right? These are truly unmet medical needs. It's not clear what the right answer is. And so that's hard for the students to deal with. So it's important, I think, for those who are embarking on this kind of a project, these biodesign courses are not like other courses. And it's, I think, useful to help the students grapple with that. Some students really just love it. They're like, wow, I get to define this for myself. Other students, they're really used to checking the boxes and they won't know how to deal with a course like this. So that's a challenge that we face. Next slide, Lynn. The challenges for us is basically the same. It's how do we help the students with the challenges that they have. And so I won't focus a lot on those. I think there are a lot of other practical challenges, and I'm looking to the Q&A to kind of be able to talk to those. Next slide. And I think we're getting to the last couple of slides here. So there are a few resources. One of them is this biodesign guide.stanford.edu. This is, I hope that you've seen this, and if you haven't, please go there, check it out. We've been working really hard to make an online version of the book, and it's something that should be accessible to people who are just getting started with biodesign, instructors, as well as students. And so this is a resource that we hope that you'll use, and we'll look forward to your feedback on it. There are a bunch of other resources that are out there. And I'm seeing questions come in, so we'll definitely be getting to those. Yeah, is there a textbook or reading materials for the basic design concepts? Yeah, thank you, Sonia. This is where I would send you first. So type it into your browser. You do have to, like, I think, share your email and why you're using it, but that's it. There's no like password. It's not password protected. All your students, and you should be able to get there. This is where I would start. And the last slide, I think, is a couple of recommendations. And what I'll do here is I'll start to go to the Q&A while you look at these different recommendations. And as people have questions, we'd love to hear them. And of course, questions for Lynn, questions for me, questions for Pao. I know we started a little bit late. I don't know if we're going to finish right in the next 3-4 minutes, or if it'll go a little bit longer. But I'll take the opportunity to thank everybody for your attention now. And then we can get to some of the questions that are in the chat.
Thank you, Roslyn and Paola. We have a few minutes to go over the time we were late, so feel free to answer the questions, Ross, please.
Okay, yeah, and I'll invite Lynn and Paola also to participate in the Q&A here. I'm just going to go from the top on these chat questions. So does the biodesign process include a business model canvas? So that is a tool that some of our courses and some of our teams choose to use in our undergrad class. We don't use that at all. But Lynn, do you want to say anything more specific about that?
No, because I'll show my bias. I think the business model canvas has some limitations in healthcare, but the process covers much of the same material, but we don't leverage it directly. Yeah, that's better said than I was going to say. You know, what are the outcomes of the class? Do the groups stay intact after the course ends, and what % follow through with their projects? So in our capstone course, we run it in the fall and the winter in part so that they can continue in the spring if they wish. And so they continue independent study with me or with another instructor. I would say about a quarter of our teams continue in some respect. Sometimes it's half, sometimes it's only one team. It's really no pressure, it's up to them. Sometimes they stay intact, sometimes they don't. Sometimes the teams will pull people from other teams to join them after the course ends. We don't do any of that during the course. We set one of my recommendations is to set the teams yourself and not to let the students switch and not to let them ever think that switching is even a possibility, because that tends to just bring drama. And the drama is not useful. And one of our learning goals is how do you work on a team? And so that's how we approach it. Other people feel differently about that, but that's how we do it. And then what % follows through with their project? So we've never had a team launch a company out of our undergraduate course. Our undergraduate course is about learning the process. Typically, their projects are interesting and exciting, but probably not worth like giving up all their other life plans to go pursue. And so I've never recommended to a student from our undergraduate course that they like not go to med school and instead launch their project. But also our course is not really designed around launching a product. It's more about learning the engineering processes around it. So some of the teams have licensed their stuff and, you know, things have continued, but not very many. You know, it's not a startup factory, it is a course. Let's see. Sorry, Ross. Oh, yeah. One thing I think that you've done really nicely with the capstone course is showing students that there are different ways to sort of get your good work out there into the universe. So often the students will write a paper based on their research. We've had, or I've used Royal We, Ross has had teams that have put their intellectual property into the public domain so that it can be leveraged by others in, for instance, low resource settings and things like that. So looking at other ways to take good works forward The other thing I wanted to comment on is we did start what we call an extension funding program for our undergraduate students. And by giving them, they can apply for very small amounts of money, like $1,000, to continue working on their project beyond class. And we found that the learning that goes on in continuing to keep the project moving without the structure of class is really immeasurable for the students. It's like next level in terms of project management, time management, learning to continue to execute on the process without the scaffolding of class has been very, very valuable, and also like a nice resume builder for students. So, and they really don't, they can apply for $1,000. Most of them don't even spend all the money, but they get tons and tons of learning through that program. So that's been a nice one.
Yeah, thank you for pointing that out, Lynn, and sharing that. And, sort of our mantra is like, the further it goes, the more they learn. So how can we support them in getting it to go as far as they want it to go? There were some questions about the clinical mentors, so I'll just say very briefly, and Yusuf, thanks for the question. Our clinical sessions are 1 hour each. We get the clinicians to sign up for 2 total hours. That's their requirement, in addition to a half hour with me before the class starts to kind of put together a story about a patient in their area. So that's kind of what we set the students up with is, meet Susie. Susie is a 45-year-old mother of 2 who works as an educator, and she had trouble climbing the steps last week. And, and sort of so we put that story together, and each team gets a different story as their starting point. And there's no particular need or project in there, right? It's like, we know that it's a fertile area. We know that the clinician cares about the area. And then I do a lot of work with the clinicians around helping them understand that this is the student's project. It's not their project. And if they try to make it their project before the end, that'll ruin the whole experience. And so we choose our clinicians, honestly, not based on their level of expertise, but on how well they kind of get it, that this is about the students running their own project. Some clinicians want students to come and work in their lab and do stuff, and that's great, but it's not a good fit for our course. So that's how we choose them. I know there were a couple different questions. Hopefully that kind of hits those few questions.
I have another question. Sorry.
Yeah, I hear a question maybe from the room.
Another question, apart from the clinical mentors, do you also have technical mentors, like an associate professor in the university that is related to the project because of the area of knowledge that is being applied in the technology development? And also because a little bit of the struggle of the course is getting the students to the lab early enough to prototype, and this is, I don't know how you manage to do it with the other professors. It's kind of pushing one of the undergrads into the research group, and that is not always easy.
Yeah, so we're blessed here to have a lab that's a sort of general purpose lab space that our students are able to use for our capstone in our bioengineering building that is different from the faculty's labs. It doesn't have everything, but it has a variety of different types of, wet lab and dry lab tools and things like that. So usually teams can get started in there. In terms of, as the projects progress, and depending upon how important specific expertise is to that project, then what we'll do is we'll draw from other people, whether it's graduate students or, like you're saying, faculty in the department who have that expertise, and we try to make those connections. And again, what we emphasize for the students is that part of the learning is just how to do that. Even aside from the technical work itself and the project work itself, it's like, how do you bring somebody up to speed on your project and get help from them? Frankly, in our best circumstance is when we get a faculty member who will have a joint meeting with the team and one of their postdocs or one of their advanced graduate students, and then the faculty member sort of disappears, and the team has direct access to a grad student or a postdoc. And then that's usually the best situation, because it enables the team to move quickly and get into the lab if they need to. And it's all over the map. Like some of our teams are like doing animal studies using a model that, you know, the clinical or the faculty mentor has all set up. And others, they just have like one meeting and they never talk to him again. So it really depends on the project. I don't know, did that answer your question?
Yeah, but that happens naturally. I mean, it's not like you put someone in their project. Okay.
Yeah, and you know, the challenge of running courses like this, as Lynn will tell you, is because every project is different, you just have to be able to kind of move quickly and kind of figure out what they need and try to help them get that in real time. That's the hard part of running this course. I like to refer to myself as like the ringleader of a circus, right? It's like an 8-ring circus where every team is a ring in the circus. And so it's fun, but it also takes that sort of commitment to being on top of things. Another thing that I didn't mention, in order to keep the pace for our first quarter, we have a 24-hour turnaround on our assignments. We give the students feedback within 24 hours of when they turn it in, which is not easy to do, but also it just works out better for everybody. So that's a comment that I'll make. And I know we're short on time. I also want to give a chance to questions for Paola or for Lynn as well.
A little bit of, I don't know if this is flirting too much with the head of the department, but what would a general purpose lab need for students to prototype? What do like if we wanted one of those, what do we need?
Yeah, I guess what I mean to say is it's sort of like it's a makerspace. You know, so we've got the 3D printer, or we've got a set of tools that somebody would have in their garage, woodworking and otherwise. We've got a whole bunch of just random stuff, tubes and fabric and, you know, all sorts of just different things to help students get their hands moving on different projects. We also have wet lab facilities. So like we've got a bench with a hood so that students who are working, whether it's with cells or with chemicals, are able to do that safely. And I'm happy to share details. And our facilities are embarrassingly nice. And I would say that the facilities, you need to have a certain basic level so that the students believe that they can do stuff. But most of the prototyping that our students do could be done like at home on their kitchen table. So it's not like the fancy equipment honestly doesn't get used that much. Sometimes it gets used, but not that much.
Thank you very much. Sorry.
I also, by the way, I was going to say, I'd like to remind the students that there's a rich history of kitchen table prototyping in medical device innovation. And include, you know, so they should think about it at that level, especially in our course. I mean, we want them to go beyond that. We want them to be making rigorous measurements, but that's where it starts. And then once it gets started, then usually, then you can figure out where to go with it. That's my experience.
And just to confirm that, Ross, during the 2 years that we were away from the campus, the students were in their homes by themselves, operated, and some nice things came. So they are capable of doing this. Of course, it is our responsibility to give better tools, and that's something that we are also quite concerned with. A better facility and better cooperation, but at home is also possible.
Yeah, and Dr. Ross, my question will be something more related about grading. Like we can separate, as in one of the slides I wrote that you separate like project from process, because like following the biodesign process does not necessarily mean that you're going to come with an awesome solution. You could often fail. However, you like somehow learn to identify, like to invent and everything. So what tips do you have, like for me as a professor or someone who is responsible for the grades, to have like some kind of measure to separate when someone may be failing or his solution may not be the best or whatever, but I see that he's like prototyping, he's making some iterations. So what metrics or what like advice would you have like to be able to separate those two aspects?
Yeah, thanks for that question. And this is something that we've only come to in the last few years, and I think it's been a big difference. And I'm glad you brought this up. I had it on the slide, but I didn't really mention it. So, and I'll just say ahead of time that this is a part that our faculty really struggle with. And in fact, most mentors really struggle with this because you want to talk about the project, right? You want to talk about what's going good with the project and what's wrong with the project. And so we very explicitly only give them scores on the process elements. We give them feedback on both process and on the project, but the scoring is only done on the process. And that's difficult because the process is squishy, right? It's a little bit, oh, well, what does it mean to get like a 9 out of 10 on a need statement? So that's something that we're struggling with. And honestly, we'd love your help as we all kind of develop our rubrics for these things. And for those of you out there who feel like you have a good rubric for one of these squishy elements, I would love to hear from you. I'm happy to share our rubrics that we use. But what we do is we very explicitly separate them. So like in our grading meetings and like all of our rubrics that we show the students, the things that score points are doing the steps of the process thoughtfully, rigorously, completely. Do you have primary sources for your things? Are the aspects of the need that should be connected? Those kinds of things. And then the project itself, we take out the red pen, or it's not really a red pen, but the project itself, each week in the mentoring meetings, it's like, okay, let's talk about the project. But then the process part, that grading and that feedback is separate, ideally. Did that answer your question? I don't know if that helps.
Yeah, somehow. Whatever is, for example, what we use is that for weekly assignments, we ask our students to make like some kind of calendar in which they establish like some milestones and some goals that they would like to achieve. And for example, it's difficult for me to grade it. He said by week 5, I'll have like interview 5 doctors, and maybe he interviewed those 5 doctors, but the questions were not meaningful, or probably he did not record any of that, or he did not make any conclusions. So in overall, he needs to succeed in his task because he went to the hospital and he talked to 5 doctors. However, can we say that that was something that added to the process? Maybe that was not the case. So sometimes it becomes difficult to deal with the students because once they see like something that is a grade that they don't like, the first thing that they come was like, here they are the 5 names of the doctors, so you have to grade me perfectly. So yeah, like somehow like to manage that, like, because at the end I think the number doesn't matter that much. It's just like the process of their learning and using it. But since students are students and sometimes they are really motivated by a grade, it would be like beneficial to have those rules clear from the beginning, so they like can do their tasks how they're supposed to do and not maybe go in the easy way.
Yeah, and in that particular case, what I would do, our rubrics will have things like the first part of the rubric is completeness. So we said to go talk to 5 doctors. Did you talk to 5 doctors? And you get scores for that. But then there would be a question about content knowledge, or there would be a rubric section on, were those questions relevant? And maybe the student wouldn't get high scores on that, right? Maybe they'd only get partial credit on that. So we like to break it down so that they can see like, oh, okay, yeah, I finished it, got full marks for that. This could have been better. This other thing I did well. maybe they reported on it really accurately, like they did a good job with the recording and they did a good job with summarizing, but like, so that's how I would say to go about it. And then that way they can see like, oh, okay, here's where I fell short. And thanks for the other questions in the chat. I think you saw answers around peer evaluation. Yeah, we have different ways of doing that. One hack I'll just share. This is one of my favorite hacks that we figured out a few years ago. Before the presentations, the day before the presentations or the morning of the presentations, we require the students to present to another team and to give each other feedback. And we collect that feedback and we give them points for having met with another team. And it has dramatically leveled up the quality of the presentations in a way that I could not have anticipated. So it's very easy to do. It requires zero effort from you, except maybe pairing up the teams. And all of the presentations, every single one, is way, way better after they do that. Another thing that we do, which I'll just share, is during the presentations, we have a rule, which is that the faculty are not allowed to ask questions during the presentations. Students have to ask each other's questions, and the faculty don't get to ask questions during the presentations. We can ask those questions in the mentoring meetings. And that changes a lot the way the students think about the presentations, because they're a lot less nervous. They don't feel like they're being put on the spot. And sometimes I have to like literally go to the faculty and like push them out of the room so that they won't ask questions. But that's another thing that I'll just share. And thank you, Lynn, for sharing about CATME, which we are using. CATME used to be free. It has now now it has a cost associated with it, but we really like CATME.
We don't use it here. We don't use CATME roles because we have to pay for it. So
yeah, CATME is unfortunately a little bit pricey, but we really like that model. I wonder if there's an opportunity there. And I just want to thank you, Matthew, for sharing in the chat. Yeah, we don't have communication experts connected with our course. That's something that's like a future thing that we'd like to do, is to connect our course a little bit better with the communication hubs on our campus. For those of you who do that, kudos, because I think that's really a great way to go.
all right, well, Lynn, Pao, Ross, I really want to thank you for the support that you are giving us. Here at Uniandes, we have been a very strong believer in the process. We have used it, we continue to use it. We have made some local modifications and adjustments that we will share with the people that are here and with yourselves. And I think that in the end, some of the most successful experiences are what the students have told us about the impact they see of their work in some patients. Those that have had the opportunity to go and try their things are surprised themselves about how simple things can make an impact. And that's something that they did not believe was possible when they started. And the fact that when they go to either continue as researchers or working for a company, this experience is always useful. This experience is always something that gives them something to talk about. So the process itself is very educational, even if they don't come up with the next startup that is very successful. As we have said here, the process of doing it is very, very useful in many ways. The ability to communicate things in a simple way is very useful. The ability to work with others, the ability to disagree, and not take it to a physical level. They are good things. So thank you for your support. Thank you for the tools. We use them and we will continue to use them. And we will see you soon.
Thank you all so much.